The rat appears at 3:47 in the morning on a street in Lower Manhattan and does something that no pest control operator, no city health inspector, no rodenticide manufacturer, and no researcher who has spent a career studying this animal has ever been able to reliably prevent it from doing. It stops. Not in the way a startled animal stops, freezing mid-movement, muscles locked by threat assessment. It stops with a deliberacy that looks on the high-resolution infrared footage the research team has been collecting for 6 months, almost contemplative. It is standing at the edge of a bait station that was placed 3 days ago, and it is doing what rats do when they encounter something new in an environment they have memorized to a degree of detail that the research team's 6 months of footage has only begun to document. It is reading the situation. Through its whiskers, which can detect air currents, fine enough to map the space around an object without touching the object. Through its nose, which is processing a chemical description of the bait station, the rodenticide inside it, the gloves of the technician who placed it, the substrate it rests on, and the absence of normal rat scent markings around its perimeter that would indicate other rats have investigated it and found it safe. The rat does not eat the bait. It turns and walks away at the unhurried pace of an animal that has assessed a situation, reached a conclusion, and moved on. By the time the sun rises and the street fills with people who will walk past the bait station without giving it a second glance, the rat is back in the burrow network beneath the building 30 meters away, where it has been living for longer than the current tenants have occupied any of the apartments above it. The bait station will sit there untouched for 11 days before the pest control operator removes it and logs the visit as unsuccessful. This is Rattus norvegicus, the Norway rat, the brown rat, the common rat, the animal that has lived alongside the human species for 10,000 years, that has followed human settlement across every continent except Antarctica, that currently inhabits every major city on Earth in populations that in some cases exceed the human populations they coexist with. It has survived every poison humans have developed to kill it, every trap humans have designed to catch it, every eradication campaign that any municipality has ever launched against it. Not because it is large or physically powerful, not because it reproduces faster than it can be killed, though it does reproduce fast, but because it is, in ways that researchers studying it have spent decades trying to fully characterize, extraordinarily difficult to deceive. The rat's relationship with the human species is older than agriculture, and is, by any objective measure, one of the most successful interspecies relationships in the history of the planet. Genetic analysis of rat populations across Asia, where the species originated, and across the global distribution it has achieved since, tells the story of a species that initially lived in close association with early human settlements in what is now northeastern China, and followed the expansion of human trade and travel routes outward across the continent and eventually across the ocean. The rat did not travel because it sought human company. It traveled because humans were carrying food, and food was where the rat needed to be, and the holds of ships and the grain stores of trading posts and the food caches of military expeditions were, from the rat's perspective, simply very good foraging opportunities that happened to be mobile. By the time European colonial expansion began carrying rats across the Atlantic and Pacific in the 16th and 17th centuries, Rattus norvegicus had already been refining its coexistence with human settlement for thousands of years. Every generation of rats that lived in close association with humans was subjected to selection pressure that favored the traits that made coexistence successful: the ability to recognize and avoid new threats, the intelligence to adapt behavior rapidly to changing conditions, the social learning capacity to acquire and transmit information about dangers and resources without individual trial and error, and a reproductive rate capable of replenishing population losses faster than those losses could be sustained. The rat that arrived in the holds of European ships in the Americas and Australia was not a naive animal encountering human hostility for the first time. It was the descendant of thousands of generations of rats that had been surviving human hostility as a standard feature of their environment. The first thing to understand about why rats are so difficult to eliminate is a behavior that pest control professionals encounter constantly and that has a specific name in the scientific literature: neophobia, fear of the new. Rats do not eat new things casually. They do not investigate novel objects without extensive prior assessment. When something new appears in an environment a rat has established as its territory, whether a bait station, a trap, a new food source, or a new object of any kind, the rat's behavioral response is not curiosity or indifference. It is caution extended over a period of time that varies with the nature of the new object, but rarely falls below several days and commonly extends to weeks. The rat assesses the new object from a distance, using smell and whisker-mediated air current detection. It observes whether other rats interact with it. It waits for chemical signals from those other rats, including the alarm pheromones that rats release when stressed or injured that would indicate danger associated with the object. Only after this extended assessment period, and only if no danger signals have accumulated around the new object, does a rat typically approach it closely enough to investigate by contact. For a bait station placed by a pest control technician, this means that even a perfectly formulated, highly palatable rodenticide bait in an attractively designed station may not be consumed by any rat in the territory for the first week or two of placement, not because the rats cannot smell it, and not because they are not hungry, but because the assessment process that precedes consumption of any novel food source takes that long in an animal as cautious as Rattus norvegicus. And during that assessment period, something else is happening that makes the situation progressively worse for the bait. The rats are learning from each other. Social learning in rats is not a simple imitation mechanism. It is a sophisticated information transfer system that operates through multiple channels simultaneously and produces behavioral outcomes in observer rats that can be as robust as outcomes produced by direct personal experience. When a rat in a colony eats something and becomes ill, the chemical signature of that illness, detectable in the sick rat's breath and body odor, is recognized by other colony members as a danger signal associated with whatever food the sick rat has recently eaten. The specific food smell that the sick rat is carrying, combined with the illness signals it is emitting, produces in observer rats a conditioned aversion to that food smell that can persist for the lifetime of the observer rat, without the observer rat having ever tasted the food. The mechanism is not learned consciously. It does not require the observer rat to reason about cause and effect. It is a hardwired associative learning system that pairs the food odor with the illness signal in a single exposure and produces an avoidance response that is essentially permanent. Researchers have documented this social transmission of food aversions across multiple generations of rats, with mother rats transmitting aversions to their pups through the same olfactory mechanism, producing populations of rats with dietary restrictions they have inherited socially rather than genetically, applied to specific foods that previous generations encountered as dangers. For a rodenticide campaign, this means that the death of even one rat from bait consumption can produce bait aversion in the surviving colony members that persists indefinitely and is passed to subsequent generations. The bait that killed the first rat is now a food the colony has collectively learned to avoid, and the learning is as permanent and as robust as a learned response based on direct personal experience. The colony that lost members to a specific poison formulation does not give that formulation a second chance. It has catalogued it through the social transmission of the experience of the rats that consumed it and became ill as a substance to be avoided, and the catalog is maintained across generations through a social learning mechanism that requires no individual rat to have personally survived a poisoning to benefit from the information. This is the mechanism behind what pest control professionals call bait shyness, and it is the primary reason that rodenticide programs that achieve partial kill of a rat population often become less effective over subsequent application cycles, not because the rats are developing physiological tolerance to the poison, but because the surviving population has acquired a socially transmitted aversion to the bait formulation that makes subsequent exposures progressively less likely to result in consumption. But bait shyness, as significant as it is not the most durable form of rat resistance to rodenticides. The most durable form is genetic. Warfarin was introduced as a rodenticide in 1948, and represented, at the time, what DDT had represented for insect control. A compound so broadly effective against the target species that it appeared to offer a reliable long-term solution. Warfarin works by blocking the enzyme VKORC1, which is essential for the recycling of vitamin K in mammals, disrupting the blood clotting cascade and causing fatal internal hemorrhaging after several days of consumption. The multiple dose requirement was initially seen as an advantage, since it reduced the bait shyness risk by not producing immediate illness in consuming rats and therefore not triggering the social aversion learning mechanism. By 1958, 10 years after warfarin's introduction, resistant rat populations had been documented in Scotland. By the early 1960s, resistant populations were documented across England, Wales, and continental Europe. By the 1970s and 1980s, warfarin-resistant rats had been documented in North America, in Australia, in Japan, and across most regions where warfarin-based rodenticides had been deployed at scale. The resistance mechanism was genetic. A mutation in the VKORC1 gene, the same gene that warfarin targets, altered the enzyme structure in a way that reduced warfarin's binding affinity without significantly impairing the enzyme's normal vitamin K recycling function. Rats carrying the mutation survived warfarin exposure that killed susceptible rats. They reproduced. Their offspring inherited the mutation. Over the decade between warfarin's introduction and the first resistance documentation, the mutation had moved from a low-frequency variant in whatever rat populations happened to carry it to a dominant characteristic in populations that had been subjected to warfarin pressure. The response to warfarin resistance was the development of second-generation anticoagulants, compounds with the same VKORC1 targeting mechanism, but with molecular structures that bound more tightly to the enzyme and required only a single dose to achieve a lethal effect. Brodifacoum, bromadiolone, difenacoum. These second-generation compounds became the primary rodenticides in commercial and municipal pest control programs from the 1970s onward. By 2003, a population of rats in Hampshire, England, was showing measurable resistance to bromadiolone. By 2010, resistance to multiple second-generation anticoagulants had been documented in rat populations across England, Wales, France, Germany, and the Netherlands. Genetic analysis of these populations revealed VKORC1 mutations, in some cases the same mutations that conferred warfarin resistance, and in some cases novel mutations that reduced the binding affinity of the second-generation compounds. The same evolutionary process that had produced warfarin resistance in 10 years had produced second-generation anticoagulant resistance in roughly 30 years of widespread use. The rat population had not learned anything consciously about rodenticide chemistry. Individual rats had not made decisions to become resistant. The variation in VKORC1 gene sequence that reduced second-generation anticoagulant binding affinity had been present in wild rat populations before those compounds were introduced, as random genetic variation is always present in large populations. The selective pressure of rodenticide application killed susceptible individuals and allowed resistant individuals to reproduce, and the resistant gene variants increased in frequency exactly as the mathematics of natural selection predict they should. What makes this cycle of resistance development particularly difficult to interrupt is the rat's reproductive rate. A single female Rattus norvegicus can produce 6 litters per year under favorable conditions, with litter sizes ranging from 6 to 12 pups. Pups reach reproductive maturity in 3 to 4 months. In optimal urban environments with consistent food availability and protected shelter, rat populations can double in size in less than 2 months. The generation time that determines how quickly a resistance trait can spread through a population under selection pressure is measured in weeks and months, not years. A resistance mutation present in 1% of the population at the beginning of a rodenticide campaign can, through differential reproductive success over 3 to 4 generations across a single year, become the dominant genetic characteristic of the surviving population before the campaign has run its annual cycle. The city that the rat inhabits is, from an infrastructure perspective, almost perfectly designed for rat survival, not through any failure of human planning, but through the consequence of human planning for human needs that create ideal rat habitat as a byproduct. Underground utility infrastructure creates a network of warm, protected, connected corridors that extends beneath the entire city and connects every building to every other through routes that no pest control program can fully treat. Sewer systems provide water, food, and dispersal routes simultaneously. The thermal stability of the underground environment eliminates the temperature extremes that kill many surface-dwelling animals in winter. The food waste generated by a human urban population provides a caloric resource deposited at predictable times and locations by human behavioral patterns that the rat population has had generations to learn and exploit. Research teams studying rat movement patterns in urban environments using GPS tracking attached to individually trapped and released rats have produced maps of rat movement that reveal something pest control professionals have long understood intuitively, but that the tracking data makes visually striking. Rats move through cities along specific habitual routes, returning to the same food sources, the same shelter sites, the same burrowing locations repeatedly and predictably. The burrow system that the colony beneath the Manhattan apartment building occupies is not a simple hole in the ground. It is an excavated network with multiple entrances, multiple exit tunnels, oriented to provide emergency escape routes from different approach directions, deeper chambers for nesting and food storage, and physical architecture that reflects the colony's knowledge of the threat landscape around it. Entrances are positioned undercover. Escape tunnels exit into spaces that provide immediate concealment. The system has been modified over time in response to the specific threats the colony has encountered, with old entrances blocked and new ones opened as the threat landscape has changed. The social structure within the colony shapes how information about threats is acquired and distributed. Rat colonies have dominance hierarchies, and the feeding behavior of colony members is not random with respect to those hierarchies. Dominant individuals eat first and eat from the most familiar, established food sources. Subordinate individuals, particularly younger rats, are more likely to explore new food sources and to encounter novel objects in the environment first. This social feeding order means that dominant, reproductively successful individuals are the last to encounter new food sources, including bait stations. The subordinate individuals that encounter the bait first function as the colony's early warning system, not because the colony has designed them to play this role, but because the social dynamics of feeding access produce this outcome automatically. If a subordinate rat eats bait and becomes ill or dies, the chemical signals associated with that illness are detected by colony members, and the social aversion learning mechanism transmits information about the danger to the colony as a whole, before dominant individuals would have investigated the bait on their own terms. The sensory world the rat inhabits is layered in ways that make the human environment simultaneously familiar in its geography and utterly different in its informational texture. The rat's olfactory system contains approximately 1,000 distinct types of olfactory receptor neurons, compared to approximately 400 in humans. This difference in receptor diversity is not simply a difference in sensitivity. It is a difference in chemical discrimination, in how many distinct chemical signals the rat can separately identify in a complex mixture of odors simultaneously present in the environment. The air above a city street carries thousands of chemical compounds at any given moment. The rat walking through that air is reading a structured chemical landscape in which each compound has a meaning, a source, a recency, and a directional gradient that together constitute a map of what has been happening in that space across the hours before the rat arrived. The smell of the pest control technician who placed the bait station 3 days ago is still present on the surface of the station and in the soil around it, decaying at a rate the rat's nose can calibrate to estimate how long ago the human was there. The smell of the rodenticide inside the station is detectable through the station's casing, identifiable as a compound not present in the normal chemical background of the territory, associated with no prior positive experiences, and associated through the social transmission of information from colonies in adjacent territories that encountered the same formulation earlier in the municipal campaign, with the chemical signatures that rats who consumed it and became ill were subsequently producing. The rat at the edge of the bait station is not making a simple binary assessment of safe or not safe. It is integrating a complex chemical picture that provides multiple independent indicators of risk, each of which alone might not prevent investigation, but which in combination produce an assessment outcome that drives the rat away before it comes close enough to touch the station. The rat's hearing extends into the ultrasonic range, with the ability to detect and produce sounds up to approximately 80 kilohertz, well above the upper limit of human hearing at approximately 20 kilohertz. Rat communication occurs primarily in this ultrasonic range, in calls that humans cannot hear without specialized equipment, and that therefore pass entirely undetected through the human environment that the rat occupies. The social information transfer about threats, food sources, and territory boundaries that the rat colony conducts continuously is conducted in a frequency range that the humans, sharing the same physical space, have no access to. The pest control technician walking through the building's basement cannot hear the alarm calls the rats are producing in response to our presence. The building's residents cannot hear the colony's social communication through the walls. The rat's social network operates in a frequency band that the human nervous system cannot reach. The whiskers that the rat sweeps through the air and across surfaces as it moves are mechanosensory organs of extraordinary precision. Each whisker, called a vibrissa, is connected at its base to a dense concentration of sensory neurons, with some estimates suggesting that a single rat whisker is connected to more sensory neurons than the entire human fingertip. The whiskers can detect air movements too subtle for the rat's other senses to register, providing information about objects in the environment that the rat has not yet contacted. The pattern of air disturbance around an object tells the rat the object's approximate size and shape before contact. The chemical composition of the air stirred by the object provides olfactory information simultaneously. The rat approaching an unfamiliar object is receiving a detailed multisensory picture of that object from several body lengths away, information rich enough to make a preliminary danger assessment before the rat has exposed itself to the risks that would come with close contact. Research conducted in the past 2 decades has added a dimension to the understanding of rat avoidance, behavior with significant implications for why rat populations maintain resistance across generations, even when the specific threats they face change. The discovery of epigenetic transmission of fear memories in rats, first documented systematically in a 2013 study at Emory University, demonstrated that rats exposed to a specific smell in association with a fire-inducing stimulus showed increased sensitivity to that smell in their offspring and grandchildren, even when those offspring had never been directly exposed to the fire-inducing stimulus themselves. The sensitivity was transmitted not through behavioral observation or olfactory communication, but through epigenetic modifications, heritable changes in gene expression that did not alter the DNA sequence itself, but altered how specific genes were expressed in subsequent generations. The implication for pest control is significant. A rat population that has been exposed to a specific rodenticide compound, and that has learned through direct experience or social transmission to associate that compound with danger, may be transmitting an enhanced sensitivity to danger-associated cues related to that compound to subsequent generations through epigenetic mechanisms, even in the absence of continued direct experience. The behavioral resistance that a rat colony acquires through social learning may be reinforced by epigenetic transmission in ways that make the resistance more robust and more persistent than social learning alone would produce. The most effective rat control outcomes documented in the research literature share a common feature. They combine multiple independent control methods applied simultaneously across an area large enough to prevent rapid recolonization from surrounding unaddressed populations. Rodenticide application combined with physical exclusion of buildings combined with modification of the food waste and harborage conditions that make the urban environment attractive to rats, applied across a defined area with coordinated timing that does not allow survivors of one method to recolonize areas cleared by another before the next method is applied. What works against rats is not a better poison. What works is removing the conditions that make the urban environment supportive of rat populations at the densities that cause public health concerns. Secured food waste, eliminated harborage, physical exclusion of buildings, applied across large enough areas with sufficient coordination, and sustained for long enough that population recovery is prevented rather than managed. The rat on the Manhattan street at 3:47 in the morning, stopping at the edge of the bait station and then walking away, is the physical expression of everything described above: the neophobia, the olfactory assessment, the social learning history encoded in the colony's behavioral library, the genetic resistance to the compound inside the station, the epigenetic priming toward caution around objects with the chemical signature the station carries. All of it in a single behavioral moment, expressed as a rat that stops, assesses, and walks away. It is not a smart decision in any conscious sense. The rat is not reasoning about rodenticide chemistry or pest control strategy. It is being what 10,000 years of coexistence with human pest control has shaped it to be. Cautious about new things, informed by its colony's collective experience, equipped with senses precise enough to characterize threats before they become contacts, and possessed of a reproductive rate that refills any population loss faster than most sustained control programs can maintain. The street fills with morning light. The bait station sits against the building wall. 11 days from now, the pest control operator will remove it and log another unsuccessful visit. Below the pavement, in the burrow that has been there longer than the current building management company has had the contract, the colony continues. The rat that stopped at the bait station at 3:47 is already asleep. It has been careful. It has always been careful. That is why it is still here.